Hello friends, welcome to Dr. Jagdish Academy. In today's video, I will discuss about the hybridoma technology. You can see this is one of the breakthrough in the molecular biology which has led to the production of monoclonal antibody. And nowadays, this is the era of the monoclonal antibodies which are used extensively in the clinical setup. So let's start with the hybridoma technology. So in this today's video, we will discuss what we mean by this word hybridoma cells. What is the methodology to form the hybridoma cells? What are the different steps involved in this technology? Okay, so starting with the introduction. So as I told, it is one of the very important technology in the molecular biology, in the biotechnology. You can see it was developed by two scientists, Milstein and Kohler in 1975. And as I told, one of the major breakthrough in the field of biotechnology, and they got Nobel Prize in 1984. And this technology is extensively used for the production of monoclonal antibodies. To see what are the monoclonal antibodies, what are their therapeutic uses, you can click the link which is given in the description section of this video. So coming down to the hybridoma cells. As you can see, the name hybrid means mixture. Oma, something related to some tumor. So we will discuss what is hybridoma cells. So we take B cells, B cells which form the antibodies. Now the property of B cell is that they have limited lifespan. Okay, they die after a certain period of time, and these B cells produce antibodies. Now we take myeloma cells. In short, we use the word M cells. Myeloma cells, these are the cancerous cells. Okay, so these have unlimited lifespan. They continue to multiply. They continue to proliferate. Okay, so they have infinite lifespan. They are immortal, but they do not produce the antibody. So now scientists, they have used the usefulness of B cell and the M cells. But how? By fusing these two cells and getting a mixture of a cell which is called as hybridoma cells. In short, we can say it is the H cell. What are the useful thing we are taking? We are taking this useful thing. Okay, it is combining the property of B and B cells. It means it is producing antibodies which is coming from this side. Second thing, it is immortal, it will not die. So that this property is coming from this side. So that means you are creating certain kind of these cells which are producing the antibodies and they are not dying. They are continuously being dividing and you are getting lot of antibodies. So this is the beauty of the hybridoma cells. So how to create these kind of hybridoma cells? That means methodology. So what are the different steps? I will just name these steps, then I will discuss all these steps in detail. Number one, immunization. That means something which is related to antigens and antibody. Immunization and collection of antibody producing B cells. Then obtaining the M cells. Then fusing these two, that is cell fusion, which is called as hybridization. Then we select hybridoma cells. What are the desired kind of the hybridoma cells? Okay. Then clonal selection and then commercial production of monoclonal antibodies. These are just the headings. I will discuss each heading separately. So first, this step, immunization. So what we are doing in the immunization? So what we are doing is we are isolating the B cells, those cells which can produce the antibodies. So we take the mouse. We take the antigens, approximately 10 to 100 microgram of antigens. These are injected in the mouse. Okay, we wait for 3 to 4 weeks. Then we re-inject the same antigen. Then we again re-inject 2 to 4 days before sacrificing. Okay, so we are actually injecting 3 times, so that lot of antibody producing B cells are produced. Then we kill these mice. Because you have exposed to antigens, so there is a lot of B cells, and you isolate the spleen. And from the spleen, you do the methylation, homogenization to get, and they will carry out this centrifugation, that is a density gradient centrifugation, for particularly isolating the B cells. Okay, so isolating the B cells from the spleen. So this is the first step. Second step is the obtaining the myeloma cells, M cells. So there should be some desirable property of the M cells, like they should not produce antibody. Then they should be immortal, they should not die. Then another very important property we will discuss later on, that in this selection procedure, they should not contain this enzyme. HGPRT enzyme should be absent. That means they should be negative. This both negative mean they are homozygous negative. This HGPRT is, H stands for hypoxanthine, G stands for guanine, P is phospho, R for ribosyl, T for transferase. That means hypoxanthine guanine phosphoribosyl transferase enzyme should be absent in these M cells. So these are the desirable properties. Then, but point is, because whenever you take the myeloma cells, they are not necessarily these negative, they are not negative. You have to create these HGPRT negative cells. How to create? The importance of using this negative I will discuss later on, but first I will discuss how to get this HGPRT negative M cells, how to isolate these ones. So what you take normal myeloma cells, okay? Then you treat with certain mutagens, that means some agent which can produce the mutation. One of the common mutagen is the ethyl methyl sulfonate, EMS. Okay, so now you treat with this one, you will create a mutated M cells. Okay, lot of mutations are there. One possibility is that HGPRT is negative, but there is a possibility that it is homozygously removed, that means both are negative, or it may be heterozygous. Okay, it may present, but not both the alleles. So that means you are having mixture of cells. But what we need this kind of these cells, negative. We need this kind of a cell, not this kind of a cell. So how to select these kind of a cells? Then what you will do? You will take this chemical, 8-azaguanine. So what will happen? 8-azaguanine will kill these cells, which are HGPRT positive. That means those M cells that contain HGPRT, they will die. Killing of HGPRT positive M cells. So you are having only these kind of these cells, negative cells. Now the point is, why does azaguanine kills the HGPRT positive M cells? So the reason is this 8-azaguanine in the presence of HGPRT produces a toxic metabolite, which is lethal to the cells that contain HGPRT cells. That means if this 8-azaguanine is added, then toxic metabolite will kill these cells, not these cells. Okay? So you selectively isolate these kind of the M cells. Next step is the cell fusion. Cell fusion is, now you are hybridizing H and M cells. You are fusing the B cells and the M cells. So this kind of the fusion is not spontaneous. Why it is not spontaneous? The fusion between the egg and gamete, which is the germ cells, is spontaneous. But the fusion between the somatic cells, non-gonadal cells, is not spontaneous. Spontaneous means of its own. So you have to do some special things to fuse B and M cells. So what are the two common methods? Number one, we use this chemical, polyethylene glycol. So you take B cells, M cells, and you take polyethylene glycol. Okay? Which will allow the fusion of these two. Plus you will add some other molecule, some other chemical, a solvent, which is DMSO. DMSO, dimethyl sulfoxide. It is actually a cytoprotective agent. It will not allow the killing of these cells to take place. Dimethyl sulfoxide is cytoprotective. Second is electrofusion. That means you take these two cells and then you apply an electric field of this much voltage, so that for a very short period of time, just for microseconds, so that B and C cells will fuse. Now, after you have done the fusion, okay, you have obtained the H cells. But now the point is whether you will take all the H cells or you take only the desired kind of the S cells. So obviously you will not take out all these cells, you want this certain desired kind of the S cells. Okay, so what is that thing? I will explain that thing. So that is the selection of the hybridoma cells. What kind of hybridoma cells are required? So I am just repeating the same thing. B cells, they were HGPRT positive. Myeloma cells, they were HGPRT negative, as I told. So when you fuse these two, hybridoma cells, they will be HGPRT positive, because that is, they are getting this enzyme from these B cells. So that means you are having three kind of the cells at the end. After you are taking the B cells, M cells, you are fusing, and then you are getting the hybridoma cells. Now you have to selectively take H cells. But how to remove B cells and the myeloma cells? So for that is what I am explaining. In a culture, B cells will die automatically because they are having a limited lifespan. Now, in the culture, now you are left with the two cells, myeloma cells and hybridoma cells, because both are immortal. So now you have to use a technique so that we can isolate H cells from the myeloma cells. So what is the principle? How to isolate H cells from the M cells? So we use a special media which is called as HAT. HAT as a selection media. Very important thing which I am going to explain. What is selection procedure? HAT, full form H stands for hypoxanthine, A for aminoterine, and T for thymidine. So now I just want to explain one thing. How the cell multiply? Cell multiplication needs DNA replication. Okay. So DNA replication is done, that means synthesis of DNA occurs by two methods. One is called as de novo pathway. De novo pathway means using the very small precursors to form the larger DNA molecules, starting from a very small molecules. But if you are using this aminoterine, which is a component of HAT media, this aminoterine will not allow de novo pathway to take place. Okay. One thing you have to remember this thing. I will explain again this point again. Then second pathway through which DNA is formed is called as salvage pathway. In this salvage pathway, there is a use of preformed purines and pyrimidines, like use of hypoxanthine or thymidine. They are converted into DNA bases. Okay, so salvage pathway. So now, but this salvage pathway will happen only in the presence of this enzyme, HGPRT enzyme. Okay, you remember this thing? I will explain in the next slide. So what we are doing? Now you see this thing? We are having the M cells, we are having the S cell, and we need H cells, we do not need M cells. As I told, there is a de novo pathway, there is a salvage pathway. So these can use these two pathways to form DNA, which is required for the survival and multiplication. But because we have used the HAT media, in the HAT media there is aminoterine, which is present in the, so de novo pathway is blocked. This de novo pathway cannot take place. So not H cell nor M cell, they cannot use the de novo pathway. What is the pathway which is left? The pathway which is left is the salvage pathway. So now M cells cannot use the salvage pathway. Why? Because they do not have HGPRT. So that is why this cannot take place. As a result, the M cells will die because they cannot use the salvage pathway because they don't have HGPRT. They cannot use de novo pathway because they are not, because that is blocked by aminoterine. But look at this H cells. They are having HGPRT enzyme. So they can use salvage pathway, so they can form DNA, so they can survive. So this is how you are selecting H cell from the M cells. They are killed. So this is what I will explain in the form of the written matter. M cells with no HGPRT enzyme, H cells with its HGPRT enzyme. So we need H cells. This is a property. You add M and S cells in the head media. De novo pathway is blocked in the presence of aminoterine. Salvage pathway can operate only in the S cells in the presence of HGPRT. So this is the procedure of selection. So S cells selectively grow and M cells die. So now there is another procedure which is called as clonal selection. As I told, we need to produce monoclonal antibodies, a single clone. Okay, single clone which can form identical kind of antibodies. Now what I am going to explain is that, you remember we took the mouse, okay, you injected with the antigen. Now the antigens, they were having various epitopes. To understand this epitope concept, monoclonal antibody, you click on the links which is given in the description section of this video, which discuss in detail about the clonal selections, monoclonal antibodies, epitopes, these kind of things. Okay. So we are having the antigens with the various epitopes that we injected in the mice. So as a result, we got different kind of the B cells. So now what is the clonal selection? Clonal selection means we have to separate all these different kind of the B cells. So because we are having different kind of the B cells, we got different kind of the H cells. So clonal selection means separating these S cells, these cells separately, these cells separately. So this is what we have to do, clonal selection. So how to do this one? So how to do? So I am explaining with the help of a written method. There were many epitopes on the antigens which were injected in the mice. Since B cells are of various types, thus S cells are of various types. So clonal selection means separation of different kind of S cells to obtain clones of S cells of the same type. That means we want to separate all the S cells of the same type. So how to do? You take a media in which different kind of the S cells are present. So you take a microtiter plate. You add cells at limiting dilution. That means you add a very dilute solution, a single drop. So that when you add a single drop, only a single cell is added. So each well in a microtiter plate gets a single cell. That means when you are adding a very single drop, in this one just a single cell is there. In this one another cell. So this is how a single cell is added in each microtiter plate. So now in all these wells you will add nutrient rich media, a media which is rich in nutrients, so that in these wells, small wells, the single cell grows and form multiple cells. So you can say, in which you are having different kind of S cells. Now in these wells you have separated all the S cells. So proliferation of S cells in a separate wells. So now another point, this was in the laboratory. So that means you got the S cells, they were of different type. Now you want to scale up. Scale up means you want to get the large amount of antibodies from these S cells. So this is large scale production of antibody, commercial production from these S cells. So this can be done by two methods. One is in vitro propagation. That means you produce in the mice, or in vitro propagation. That means you carry out the fermentation either in a suspension culture or in an encapsulated cell. So we will discuss each one by one. First, in vitro propagation. So what we take? We take the mice. So first of all, you take a pristine. It is just a, you can say alkane, type of alkane, long chain alkane. So you inject 0.5 ml in the peritoneal cavity so as to increase the fluid content, swelling. Then you add hybridoma cells in the peritoneal cavity. So now tumor cells will grow. Okay, so as the tumor cells will grow, they will start secreting antibodies. So all the antibodies are accumulated in the peritoneal cavity. You kill the mice, remove the peritoneal fluid, then you can purify the monoclonal antibody from this peritoneal fluid. But the major limitation is that you have to kill lot of mice. You have to take lot of mice. An example, if you want to produce 1 kg of monoclonal antibody, you have to kill more than 3000 mice. Okay, so this is in vivo propagation. Second is in vitro propagation. We can proliferate these cells in the in vitro, in a fermenter. Okay, fermenters, which are also called as bioreactors. Now we can use two kind of fermenters. One is called a stirred fermenter. That means a fermenter in which you put a stirrer. Stirrer moves the cells and the media so that the cells remain in a homogeneous condition. Stirred. Okay, so you use a mechanical stirrer. So you mix the culture and the cells. But you have to remember that these are the animal cells. And animal cells do not contain the cell wall. So when you stir these cells with the help of mechanical stirrers, so these cells will be destroyed and cell will not survive. So this is not a preferred method actually. So what is the preferred method? Airlift fermenter. What you are doing is, you are not using the mechanical stirrer, you are using the air to mix the cells and the culture media. Okay, so airlift fermenter is particularly used. So coming down to the how to take cells, how the cells can be maintained in the culture media. We can use the suspension culture. That what we are doing is, you take these cells, suspend in the culture, use the airlift fermenter. More cells are growing, they are multiplying, they are secreting the products in the culture media. Okay, the monoclonal antibodies are released in the culture media, and at the end you take out the culture media, remove the antibodies, you carry out the purification using the chromatography or other methods. Second method, but we can use some other method also. Not as simply, you cannot, in this method you are suspending the cells, but in the second thing what we can do is, you can use the encapsulated cells. That means hybridoma cells, they are surrounded by the semi-permeable membrane, which is a polylysin membrane. So what is the benefit of this kind of thing? Cells, when you surround the animal cells around a membrane, polylysin, they become more stable. Okay, so you can use the mechanical stirrer also. So stirred fermenter can also be used if you use the encapsulated cell. Another benefit, because you have surrounded the cell with the membrane, the product will be retained within the encapsulated cells. They are not secreted in the culture media. As a result, the purification becomes much easier. Extensive purification is reduced. So in the upcoming video, I will discuss about the what are the diagnostic applications of the monoclonal antibodies. Thank you.